As Dr. Hutchinson said, I'm Doug Goodin. I'm from the Department of Geography here on campus. And I've actually kind of been affiliated with the NRES program for a little while now, so it's kind of interesting to give a presentation for you. What I want to talk about this morning is kind of related to in a lot of the research that I've done during my career. It's related in many ways to this notion of remote sensing, but it's a little bit more than that. It's, in a sense, another way or an additional way of kind of thinking about the data that we can get through remote sensing. So this is something that's come to be called environmental spectroscopy. And, you know, a lot of this notion of being able to gather information from a distance has become really familiar to us. It first kind of entered my conscience this way. I was a big Star Trek fan when I was a kid. That probably doesn't come as much of a surprise to anyone. Back when I was a kid, this was Star Trek, right? Most of you, if you're familiar at all, it's probably the thing on the right there. But they had this little marvelous device called a tricorder, right? And all they had to do was point it at something, and you know, it made this little whistling noise, and then it gave them information about it. And I was always really just fascinated by that. And they had bigger versions on the ship that they would point at the planets, and that to me just seemed like a really interesting and incredible thing, that you didn't actually have to be able to be in contact with something to get information about it. And as I kind of started my career, this notion of being able to use sensors that passively receive some kind of information from the environment and then convert it, or passively receive some kind of signal from the environment and then convert it into information has always remained really, really interesting to me. So this is kind of where my interest in all of this lies. So what I want to do today is a few things. First of all, I want to talk about this notion of what a spectrum is and what kind of information that you can get from one. Because this idea of analyzing patterns of reflectance or transmission in some part of the electromagnetic spectrum is not in any sense a new one. It's been around for quite a long time. You might argue that it's something that we human beings have been able to do ever since we evolved this, you know, capability to detect light with our eyes. So I want to talk a little bit about that. I also want to think a little bit about applications of spectroscopy in the Earth system, which is the particular interest that I have. And then finally, I just want to look at a couple of examples of the types of things that I've done in research and that we might be able to kind of do as part of an environmental analysis investigation. All right, so here's a bunch of questions. And they might not necessarily on the surface seem all that closely related to each other. I mean, what are stars made out of? There was, you know, reputable scientists as late as about 1929 that would say you really will never know that, right? And yet we do. We have a really good idea about what the elements that make up stars are, even though, you know, the closest we ever get to one is several tens of millions of miles. You know, is there the right amount of stuff in chocolate bars? That may seem like a really trivial question, but there's actually really important legal implications to that. If you sell chocolate with a certain kind of label, it has to be composed of a certain kind of stuff. Well, how do you know without melting it? How can you tell ranchers where the best grass is for their herds? We'll look at some examples of that a little bit later on. How can you tell if there's water pollution without actually going out and scooping a little sample out of the lake? How can you tell if a crop has a disease? What is the surface of Mars made out of? Again, these may all seem like not very closely related questions to each other, but in fact, they really are closely related to each other because they can all be addressed in one form or another by collecting reflectance or transmission information in various parts of the electromagnetic spectrum and then analyzing it using this kind of general approach that's referred to as spectroscopy. So what I want to talk about, it kind of touches on a lot of these, but what I'd like to kind of have you understand is that this is part of a much bigger type of question or a much bigger approach to answering questions like this. So what do we mean by spectroscopy? And again, this is something that's not especially new. It's an idea that's been around for a long time. And in the simplest possible terms, you could say that basically we just look at light interacting with substances as a kind of an analytical tool. And again, if you want a sort of an intuitive grasp for this, you have a very good light collecting apparatus already built into your body, right? You've got these organs on the front of your head that collect electromagnetic spectrum radiation in certain parts of the spectrum. And you've got this analytical tool that you were born with that helps you kind of understand that. So in a sense, we humans kind of do spectroscopy. We can tell something about the nature of objects simply by looking at them. Spectroscopy kind of takes this idea, it extends it to parts of the spectrum that the human senses aren't really able to reach, and also kind of applies some more quantitative and repeatable analytical tools to this. So in a sense, this is kind of what we're doing here. We're just kind of looking at the interaction of light. So one question that kind of comes to mind if you're thinking about looking at interactions of light is, what do we mean by light? And this again is probably something you've had a little bit of experience with. At some point in your life you've probably had a physics class or a chemistry class or something where you talked about the fundamental nature of light. And if you want to understand the notion, if you want to understand the ideas of spectroscopy, it's kind of important to kind of fundamentally understand what light is. So light is, more properly, it's electromagnetic radiation. The best way probably to define it is that it's energy that has a couple of properties. It propagates in a certain form, it propagates in the form of a sine wave, and it's capable of propagating through a vacuum, and it travels at a characteristic velocity, about 300,000 kilometers per second. It's emitted by any object that has a temperature greater than absolute zero. So anything that has an object greater than absolute zero is going to emit somewhere in the electromagnetic spectrum. So here's a nice little diagram. You see these in books all the time. They show this kind of, nice wavy pattern. I still have a really hard time understanding what that actually means. I kind of, at this point, have sort of given up on really understanding it. But I can kind of think about the properties of it. So probably the most important property that we use in spectroscopy is the notion of wavelength, the distance between two of those crests or two of those troughs. And the shorter the wavelength, we kind of measure that wavelength. And we use that wavelength then to kind of arrange electromagnetic spectrum radiation on this thing called the spectrum. Now, when we say light, what we usually mean is visible electromagnetic radiation, right? The stuff that our eyes are sensitive to. So your eyes have these little cells in the back of them that are capable of responding to this. But that's really just a tiny, tiny little slice of the spectrum. This kind of brownish bar you can see up here represents the entire spectrum. And you can see it goes from fractions, billionths of a kilometer, of a meter wavelength, all the way up to meters. So essentially kind of the same phenomenon, just with vastly different wavelengths. Visible light is that tiny little piece that's kind of right there in the center, basically. That's an important one, though, in spectroscopy, and it's an important one in human eyesight as well. And as probably would come as no surprise, it's also one that the sun puts out a lot of. So our eyes have sort of evolved to take advantage of the ambient radiation that's in our environment. So we're going to talk about light, or talk about electromagnetic radiation that's in several parts of that spectrum. And one of the real advantages that we get from spectroscopy is that we use devices that can detect this radiation in regions of the spectrum that are very different from what our eyes are. They're longer or shorter than what our eyes are capable of. So in a sense, it kind of extends human vision out to parts of the spectrum that normally we wouldn't have any access to. So I've used this term spectrum a lot. So just by way of introduction, I guess, might as well kind of think about what a spectrum is. And it's really just a pattern. Sometimes these things are referred to as signatures. That's a term that's kind of loaded a little bit. I personally try to avoid using it to the extent that I can. But essentially, it is just a pattern of reflectance across wavelengths. So here's a type of spectrum, or here's a type of signature, if you will, that is a really important one if you're doing terrestrial remote sensing. This is the reflectance pattern. This particular one is green grass, but it's the reflectance pattern of healthy photosynthesizing vegetation. from wavelengths that vary from about 0.4, which is blue light, basically. It's just the shortest wavelengths of light that your eyes can detect, up through about 0.7 micrometers, about right here, which is the red light. It's the longest wavelength of light that your eyes can detect. And then all this stuff out here is various parts of the infrared, parts that your eyes can't see, but can be detected using various kinds of devices. And what you want to notice here is that there's lots of little bumps and wiggles and kind of squiggles in this curve. And those bumps and wiggles and absorption and reflectance features, they're manifestations of the way light interacted with, in this particular case, this green leaf. And they give us information. If we know how to kind of decode this pattern, if we know what these various little absorption and reflectance features mean, they give us physical information about that leaf. And as we're going to see, they can tell us quite a lot about the condition of some kind of vegetation. So this essentially is what a spectrum is. Spectra come in two different types. Actually, there's probably more than that, but I kind of deal with two different types. There's reflectance spectra, which is what we just looked at. And you can, again, kind of think about that as what the pattern of reflectance off of something is. And then there's also what are called transmission spectra, which is what happens if you pass a beam of light through something. All right, so here's a kind of an example of a transmission spectrum. This is actually a transmission spectrum for polyethylene film, basically like strand around, right? And again, you notice that there are these places where almost no light passes through that thing. And that probably comes as a little bit of an intuitive surprise, because if you just look at a piece of polyethylene film, it looks transparent, right? And yet there are regions of the spectrum where the light is not passing through. So something about that, there's some kind of fundamental physical process that's going on there that's blocking that. Just like there's some fundamental process in that reflectance spectrum that's causing certain areas of the spectrum to reflect more than others. And the whole kind of science of spectroscopy essentially boils down to detecting where those reflectances or where those transmissions or those absorptions are in the spectrum, and then trying to understand what they mean, basically. All right, so just a little bit more about light, I guess, because again, if we're going to be thinking about environmental spectroscopy, about processes that take place in the Earth system, we want to sort of narrow the field a little bit. We're not going to worry about certain kinds of processes because they're really not going to be that relevant to us. What we do want to worry about is particular regions of the spectrum that are present in the environment and capable of kind of giving us some kind of information. Maybe it'd be better to say we can derive certain kinds of information from them. So nearly all of the light that we're going to use, not entirely, but nearly all of the ambient light, the illumination, the environmental illumination, comes from the sun. Which, again, we don't want to kind of get too deeply into what the sun actually is, but it's this ball of plasma, basically. And it's really hot, right? It's far hotter than anything in the Earth system. So it's radiating energy out into space in a fairly broad region of the electromagnetic spectrum. Has a temperature, a radiative temperature of probably somewhere on the order of about 6,000 kelvins. And kind of back in the late 1900s, first part of the 20th century, this guy, anybody know who that is? That's Planck. He figured out that, and I just kind of came to this conclusion, I guess, sort of theoretically, that an object of that temperature has a particular distribution of light that it emits, right? And so this has become kind of the fundamental physical law that we use to kind of understand the presence of this radiation, this electromagnetic radiation in our environment. So this is actually that curve that you can see on the upper left there is the spectral radiance curve for something like the sun. And you can see that it's emitting from relatively short wavelengths, shorter than visible light, ultraviolet, for example. So if you go outside on a nice, hot, sunny day and leave your skin exposed, you'll quite likely get a sunburn. That's the result of ultraviolet radiation, which you can't see landing on their skin. There's a big hump right here in the visible part of the spectrum, which again kind of makes sense, right? We've evolved these organs to take advantage of that. And then out through the various infrared parts of the spectrum. So I just basically show you this to kind of point out the fact that we don't just kind of arbitrarily use certain parts of the spectrum. We're using what's available in the environment to try to get this information. And we also kind of have to come to terms with the fact that although the sun is emitting all this light, all this energy out into space, some of it is kind of intersected by the Earth, right? Not all of it gets into the Earth system, and that's actually a good thing. There are regions of the spectrum that do not penetrate or don't penetrate very well through our atmosphere. Probably one of the most important ones of those is ultraviolet. Most of the ultraviolet, or a lot of the ultraviolet actually gets blocked in the upper part of the atmosphere, and that's good. If that wasn't happening, then, I don't know, it may not really be possible for life to evolve the way it does. It would just simply be too burdened by this relatively powerful radiation to really evolve. But there's also little parts of the spectrum that would be really nice to have, but we don't. So there's regions that are absorbed by ozone, like carbon dioxide. Big issue right now, right? We're sort of through emission of burning of certain types of fuels, kind of plugging that hole up a little bit, and that's not necessarily a good thing, but that's really not the topic for today. But the point here is that we're limited a little bit in the parts of the spectrum that we can actually use by the nature of our atmosphere, basically. So what's left behind then is available for us to take advantage of, to use as an analytical tool. So we kind of have to think a little bit then about what happens when light kind of bounces off of something. And again, I use bounce off here as a kind of metaphor more than anything else. It's really not bouncing off of it in the sense that a rubber ball bounces off the floor or something like that. But it's, as a kind of a first approximation, it's not a particularly bad way of thinking of it. So what happens when light hits or interacts with something? Well, and as we've seen, generally it doesn't interact with it in a very uniform way, right? And one of the real intuitive ways to kind of think about this is just to look around at the environment. And if you look around at the stuff around you, it all, or a lot of it looks like it has different colors, right? I mean, you're sitting in seats that have purple backs and you can tell that they're purple. So that tells you that it's reflecting more red and blue light, and it's absorbing greens and other colors a little bit more, right? So there's a pattern to that reflectance. It just happens to look purple to us. And again, if that weren't the case, then our environment would not exist in colors. It would just exist in shades of gray. So those colors are kind of an intuitive idea of what happens when you get differential reflectance off of something. So what causes all of that? And that's where this can all get a little sticky, basically, because how all that works, what causes things to be absorbed or reflected, gets pretty deeply into the kind of physical and chemical properties of substances or elements. So I don't want to get too deeply into this, but it's kind of good to kind of at least a little bit of an understanding of what's actually causing all of this. So basically what happens is, and again, this is a thumbnail sketch of something that's oftentimes an extremely complicated process. What essentially happens though, depending on the wavelength, is that certain wavelengths of light will be absorbed in the process of driving some sort of a change in either an atomic or molecular system. In the case of atoms, what typically happens is you get all those electrons, all those little particles that exist in a swarm around the nuclei, become excited, right? They go up to a higher energy state. Well, something has to make that happen, right? And it's just absorption of energy. But that absorption takes place in a really often very specific parts of the spectrum. For longer wavelengths, a somewhat similar process often happens, but it's on a molecular level. So molecules, of course, are more than one atom bonded together. And they can do, they can kind of respond in various ways to the presence of electromagnetic radiation. They can stretch, they can rotate, they can flex again. I don't know, anybody ever take a chemistry class where you had to build models of molecules? You know how some of them were like put together by little springs and things like that? Well, that wasn't just, you know, an arbitrary process. There was a reason for that. So all those sorts of dynamics actually kind of operate to determine at what wavelengths and how much light is actually going to be absorbed. And again, this is, as you can probably imagine, extremely complicated process. I have a really tenuous grasp on it at best. But you can kind of think about it as happening in certain wavelengths, and it sort of helps us to understand the wavelengths that we're really going to be using here. So most of those atomic processes happen in shorter wavelengths, a little bit in visible light, but mostly down here in the shorter ones. The ones that we mostly consider in remote sensing and environmental processes are more molecular processes. So a lot of those absorption features and reflectance features we're going to be looking at here are the result of kind of dynamics of various molecules. So again, just to kind of summarize this, if you look at the little bottom here, infrared primarily acts on molecules and vibrations, basically. Okay, so that's a real kind of quick and very quick look at the sort of physical properties of all this. Fortunately, we really can kind of, we don't necessarily need to examine things at quite that level of detail. So that's kind of what makes a spectrum or what causes one. But how do you actually detect a spectrum? Well, basically what you have to do if you want to make a spectrum, if you want to break light down into a spectrum, is you got to make it bend. So many of you at some point in your life have probably played with a prism, right? And what happens when you take a prism and pass a beam of light through it is it'll make a nice little rainbow pattern on the wall or something. And what's causing that is the fact that when you pass that light through that glass, that kind of triangular piece of glass, it bends. And different wavelengths bend a different amount. So shorter wavelengths bend more and longer wavelengths bend less. So you end up with kind of breaking the light down into its various constituents. So here's a nice spectrum resulting from a prism. You can see the shorter wavelengths are down this end, the longer wavelengths here. You can't see it with your eyes, but if you could use some sort of instrument that would detect infrared, you would see that there are these other colors out here, basically, these other parts of the spectrum. So you can make a spectrum by bending it with a prism. The problem with it is that it doesn't really bend it very well, or not necessarily the right way. So it doesn't bend it very with much resolution. So in practice, most of spectroscopic analysis is done through the use of what's called diffraction grating. And diffraction grating is basically just a little piece of optical glass that has had a bunch of very, very, very thin lines cut into it. So literally tens of thousands of them. And all those lines, when the beam of light interacts with each one of those lines acts as a separate little reflector for light at a particular wavelength. So the result is that it breaks the light down into the spectrum in much the same way that the prism does, but in a lot more detail. And in fact, I bet you all have at least one pretty good diffraction grating in your, if not in your possession, then at least you own one. Have you ever taken something like a DVD and sort of held it up to the light? You notice how it kind of makes a little rainbow, basically? Well, basically what a DVD is, right, is it's a surface that's been etched with a laser that contains that embeds a signal on it. But that process of etching produces something very similar, in fact, essentially identical to a diffraction grating. So if you actually, I would urge you to try this sometime. If you actually peel the coating off of a DVD, it becomes transparent, and you can use it as a diffraction grating. In fact, I'll show you a little bit later on. You can make your own, I was going to bring one with me today, but I couldn't find it. You can make your own little homemade spectrometer out of a DVD. So we'll talk about that a little later on. So, why why is it then that we don't use prisms? Why do we want to use diffraction gratings? I mentioned it was because of the resolution of it. And by resolution, we basically mean the detail with which we can break down that spectrum, the kind of fineness of the spectral features we can see. And what we're often really trying to do when we do spectroscopy is we're essentially trying to find what are called spectral lines or absorption features or reflectance features or something. But they're just discrete regions of the spectrum where energy has been used. So remember I showed you that picture of Max Planck and his curve a little bit earlier. And that wasn't just a history lesson, there's a reason for that. If you think about that curve, right, it's a continuum. All those wavelengths are present. But if you then interacted that beam of light with some object that is going to begin absorbing or reflecting various regions, those absorptions or reflectances are going to happen in really discrete parts of the spectrum, little identifiable wavelengths. So we call those spectral lines. Those spectral lines are just regions of the spectrum where particular molecules or particular substances, which are made out of molecules, are known to absorb. And the whole deal with spectroscopy is basically just analyzing those spectral lines, figuring out where they are and identifying them based on where they are, and then looking at how much they reflect, or maybe their shape, which can give you a lot of really important physical information about the object in question. So here's this diagram on the right here shows spectral lines two different ways. This is kind of a representation of them as sort of the analog, right? It's essentially someone has taken a picture of the spectrum. This used to be about the only way of doing this, but now you mostly see them like this. So these are the same thing, right? These are the same spectrum. This one's just in an analog form. This one's more in a digital form. So we're mostly going to look at spectra that look like this, but you want to kind of understand that this is sort of an analog version of the same thing. These three things here, this is just a spectra that I found online. These are all spectra of lights, CFL lights, right? What does a CFL stand for?
Compact fluorescent.
Compact fluorescent, there you go. Yeah, these are all the spectra of compact fluorescent lights. So the yellow one there is an IKEA CFL, so if you ever go to the IKEA store, you probably have seen these. But you can see they all have a pattern. And if you understood enough about the chemistry or the physical properties of compact fluorescent lights, you could just look at that and say, oh yeah, that's a compact fluorescent light. So spectrometry then, as opposed to spectroscopy, which is sort of the general study of all this, spectrometry, the measurement of spectra then, kind of boils down to looking at that pattern and interpreting that pattern qualitatively, but also quantitatively. So here's the basic idea of absorption spectrometry, right? An entire, an unaltered distribution of light passes through it. It goes through something. Some of it gets absorbed, and those regions that get absorbed then are spectral lines. Okay, so that's, again, kind of a whirlwind tour of what spectroscopy is, what spectrometry is, and how it works, and the type of information that it ultimately kind of yields for us. Well, if we want to do some sort of application, particularly in an environmental context, what we really want to do then is understand how to interpret the patterns of those absorption features or reflectance features, both qualitatively to try to, as an identification, and quantitatively to tell us something about the physical or the biophysical nature of it. So in my field, this whole notion got its start in the analysis mainly of geological features. So this is a kind of a classic example of that. These spectra here represent kaolinite minerals, and they're identified by this pattern of spectral reflectance right through here. So if you get this really strong absorption feature right at about 2.21 micrometers, 2.21 millionths of a meter, it's a really strong indicator that this minerals from this particular group are present. And this has been used for mapping mineralogy, for example, of a number of regions, basically. This particular one became really important because these kaolinite minerals were often really closely associated with economically valuable deposits, basically. Something that you could make money off of. So there was a lot of real attempts to map this kind of thing. So this is where a lot of the beginnings of using spectroscopy in the Earth system really came into play. Of course, the problem, the really good thing about doing minerals is a mineral is pretty much a mineral. Biological stuff, which is what the field has kind of expanded to also encompass, is a lot more tricky because living things can manifest themselves in a lot of different ways. So here's kind of the classic example of that. Right? This is actually a chlorophyll absorption spectrum. Chlorophyll, of course, is this molecule that allows plants to turn sunlight and water into sugars and carbon dioxide. Sunlight and carbon dioxide into sugars and oxygen. And it's probably, when you think about it, maybe the most important stuff on the surface of the Earth, right? Because ultimately, almost everything we eat either was a plant or ate something that was a plant that got its energy from this photosynthesis process. So it's kind of crucially important to be able to understand the distribution of this on the Earth system, but also how it kind of works. And basically how it works is it absorbs light in two parts of the spectrum, right? It absorbs light down here in the blue part of the spectrum. It absorbs a lot of light up here in the red part of the spectrum. It doesn't really absorb much green light, which means what? It's reflecting green light, right? So if you go outside and look at some green, the vegetation looks green because green is not being used very much in that photosynthesis process. So this is the basis for a lot of spectroscopic analysis of plants and vegetation material. And it's a lot more tricky, unfortunately, for us. So I mentioned that spectroscopy, or the spectrometry, this use of spectral information, has been around for a long time, and it has. And it's also been used in a lot of different, for a lot of different scientific applications. Some of the earliest usages of it in what we might now think of as spectrometry in the Earth system came about in biology or biochemistry or analytical chemistry of various types. But the thing that kind of distinguished it, a lot of that, is that it was, in order to do it, it was necessary to bring the sample, whatever it was you were trying to analyze, into a lab, essentially. So these are bench spectrometers. There's various types of, a couple of them that are, but they're characterized by the fact that they need to be used in a laboratory setting. And that's really powerful. They can be used for a lot of things. However, things are often different in a lab. Even the same materials will behave differently in a lab than they will in the environment. So finally, this kind of brings us to this notion of environmental spectroscopy. And what environmental spectroscopy is really all about is, instead of bringing the sample, this thing we want to analyze, into the lab, we kind of take the lab to it. We take the instrument to the sample. So here's a couple of pictures that sort of illustrate this idea. One of the things that's made this possible is through various kinds of technological advances. We can now take pretty powerful, pretty capable spectroscopy equipment out into the field with us. So that one in the upper left, I don't know how well you can see it. There's a lot of glare on the screen, but there's this backpack thing that I'm wearing here. Basically has a really, really capable spectrometer inside. Actually has three very capable spectrometers inside of it. So I point that little fiber optic, in this case a vegetation canopy, it collects a signal, breaks it down into that spectrum, records it, and we can then take the information back and analyze it in various ways. This thing in the center here is the same basic idea, but they're doing some kind of field spectroscopy over a tree. So you've got to get higher up, so we got this lift, this cherry picker thing. And on the lower right is something that is becoming much more important in all this, the use of little drones, little UAVs, UASs, small, uncrewed, free-flying aircraft that carry some sort of sensor along with it. And environmental spectroscopy also is kind of a combination of field and lab analysis. So a lot of groups that are working in this area, and again, not a necessarily new idea, although the term is relatively new, but the group that I did my my doctoral work with, which was late 80s, early 90s, we were taking approaches like this even then. We were doing, in a sense, laboratory analysis along with the field analysis to try to use both to sort of understand the nature of what we were observing through these instruments. So here's a kind of an illustration of that. This is a sample being analyzed in the lab, a similar analysis being done. I don't know whether you can see it, but you can see that spectral reflectance curve on the screen there. They're measuring it from a leaf sample right there. So this is a kind of illustration of how this is actually done. Okay, so when I first kind of introduced this, I also used this term remote sensing, which again is not new. It's a relatively well-established idea, but it's really, I think, been linked together nicely with environmental spectroscopy. And in fact, to the point where it's really sort of hard to disentangle them anymore. But remote sensing is this idea that we collect electromagnetic radiation from some sort of platform, some sort of instrument mounted on a platform. We most often, at least in my case, when I hear the term, the first thing that really pops into my mind for the most part is satellite-based platforms, but that's certainly not the only type. So one of the real powerful combinations of ideas or techniques, I think, is the combination of remote sensing with environmental spectroscopy. So here's a nice slide that sort of shows a lot of remote sensing satellites. Some of these are not necessarily imagers in the sense that we think of, or that we normally use, but there's a lot of them out there, and there's becoming even more. In a sense, we're now kind of in the golden age of satellite remote sensing. What'd you say? Yesterday they launched the second central satellite, right? There are plans. I just made a real quick informal count this morning, and probably somewhere in the order of about 50 remote sensing satellites will be launched in the next year to year and a half. That's 5 to join the, I don't know, probably close to 100 that are up there now. So, it's kind of becoming sort of crowded skies in some ways, but it sort of illustrates the development of this technology. In addition to these satellite platforms, there's aircraft-based platforms that are in common use now. I like this picture right here. This is actually an entire airplane. This has got a little sensor package on it. You can see it right there. That was basically developed from the ground up as a remote sensing platform. A lot of its intended applications of military, but it's also being used in some civilian applications as well. So obviously that's not something that you're going to buy on a beer budget. That's several $1 billion worth of aircraft and sensor there. But it kind of illustrates one other approach here. This one on the left here is also an airborne platform, but kind of on a different end of the scale. This is basically a rather run-of-the-mill fixed-wing aircraft, and they're essentially temporarily mounted a sensor on it. So this just illustrates the idea that airborne platforms can run kind of a continuum. All right, so what's the difference then between, or what distinguishes spectroscopy in the remote sensing field from other ways of thinking about it? And it basically boils down to the type of signal you're getting. Most remote sensing instruments, and even most of the ones, if you've had one of my classes, for example, most of the stuff we talked about in the class was really just kind of broadband multispectral data, great big wide bands, distributed discretely throughout the spectrum in places that are kind of important for various kinds of information. Spectroscopy, on the other hand, involves, instead of just selecting regions of the spectrum and having really wide bands, and just having a few of them, 5 or 6 or 7, it involves hundreds or perhaps thousands of extremely narrow channels that are all contiguous with one another. So it gives us complete coverage of the spectrum. So the term that's most often used for that is hyperspectral remote sensing. So in some sense, you can think of environmental spectroscopy as being related to hyperspectral image or hyperspectral remote sensing. So we end up with this thing we call imaging spectroscopy. Imaging spectroscopy is essentially imaging in 3 dimensions, I guess you could say, rather than just pointing a sensor at something and collecting a reflectance pattern. We collect a whole series, a whole grid of reflectance patterns distributed in this XY space. So here's this nice illustration of that. Each one of these pixels in this image, if you tunnel down in the reflectance direction, is itself a spectrum. Here's the form of data that has often come out of this. It's called a data cube. The data cube basically just kind of shows you the 3-dimensional nature of this. And in my classes, we have a lot of fun making data cubes and then like playing with them, spinning them around. So we got various angles and slicing and all kinds of stuff like that. But they're not just pretty pictures. There's actual information embedded in it. So it's a way of kind of visualizing this 3-dimensional data. All right, so just to kind of recap, talked about this idea of what a spectrum is, what spectroscopy is, what remote sensing is, how environmental spectroscopy can be kind of linked together or married up with remote sensing to, in a sense, help us analyze problems or processes or questions about the Earth system. So I wanted to give you one example. And this is an example that is actually some research that we've done here. It's one of my doctoral students actually did a lot of this as part of her master's. But I think it's a pretty good illustration of the types of things that environmental spectroscopy can and is being used for. So the idea, the central thing we were interested in here was not directly necessarily a grazing process, but what we were interested in was can we measure the distribution of nutrients and protein and other grazing-related properties of the canopy. And this is actually this is not just a trivial problem. Grazing is a huge part of the economy of the state. In fact, in this particular region, it's extremely important. Be able to map where good forage is, where good grazing is, would certainly support that grazing economy. And it also is important from an ecological perspective, or from a management perspective, to be able to use this to kind of detect where and how much, what the potential for grazing might help better manage this kind of a resource. So we did this particular work by reflectance spectroscopy. We come back here to this, I showed you this earlier, right? And I said we kind of come back and look in a little bit more detail at all these little bumps and wiggles here. And actually, what we ended up analyzing, this I think is a really good sort of object lesson about environmental spectroscopy. You know, if you look at this, you see there's all these kind of large features. There's a big absorption feature there caused by water. There's one there caused by water. There's one there also caused by water. This feature right here is associated with the amount of chlorophyll that's in and the structure of the cells that are in the leaf. But the thing we looked at here is, and I hope you can see this. You see that little thing right there? I mean, if I cover it up with my finger, you can't see it. There. That is actually an absorption feature that is associated with chlorophyll. In fact, it's a lot more cleanly associated with chlorophyll than any of these two right here. So we use that little subtle feature at about 670 nanometers to quantify the amount of chlorophyll, which is closely related to nitrogen that's in the canopy, which is nutrients related, protein content, right? So that's what we were kind of looking at here. We did this research out at the Konza, which some of you are perhaps familiar with. The nice thing about the Konza is that it's this, number one, it has a captive grazing population, bison in this case, and cattle. And it also has, it's also been treated in a way that produces a wide variety of conditions. Right? All these little experimental units have been burned at different intervals, they've been grazed or not grazed, and you've got all this nice topography out there. So it produces a lot of variation, and it gives you a nice kind of challenging environment with which to see if you can extract the kinds of signals that we would need to be able to map, for example, the distribution of nutrients. We did this with a combination approach. Basically, we used a combination of field measured spectra. So that's not one of us up there. That's actually one of the people from the company that makes that instrument. And he's just he's just a little bit to the west of Boulder, Colorado there, which is where they make these things. So you can see a little further back, you'd see the flat irons, those flat rocks that are above Boulder. But anyway, he's doing essentially what we did, though. He's carrying this spectrometer around, pointing it at various surfaces, and collecting spectrum. We also had an aircraft overflight of 4 times during the 2013 growing season, where we collected similar sort of spectra, but in the form of those data cubes, right? Imaging spectroscopy. And we used all those together to analyze the spectra. So we use that fine absorption spectra, like I said, about 562 to 600 nanometers. Right about there, if you can see that. And this is what we came up with. This is actually a map of the amount of nitrogen in May of 2011 across a lot of the Konza. You can see there's a lot of variation in this. It varies from very low amounts into some cases to relative very time. I notice people are starting to look in the window. So again, this thing can be used to kind of help understand the grazing behavior. It can also be used as a way of understanding the distribution. Okay, so that was one example. One of the kind of new directions that people are doing is these use of little drones, little small unmanned aircraft to do this. Here's one that I think is really cool, and this is what I'll kind of close off with. Spectroscopy has become kind of citizen science. You can actually build a little spectrometer. This is what you would do with a little piece of a DVD. Right? You peel the DVD off, you put it in this little box, you put a little camera behind it, one of those little cameras that goes in your computer, plug it into your computer and you get spectrum. And there's a version that goes on your phone. And people are actually using these to make measurements in a variety of places. Here's some guy, I thought this was interesting because he's basically dressed in a suit and tie, making a spectroscopic measurement, obviously, in an urban environment. Here's some people that are monitoring river quality in Hopewell, Virginia, using instruments very similar to the one that I just showed you there. And they're looking for, in some cases, very hazardous chemicals that are in the water. So this is kind of an example of environmental spectroscopy sort of liberated by the people, in a sense. And this is not just a trivial thing. You know, the state of Wyoming has kind of made it difficult, if not illegal, to collect environmental information about water quality. So here's an example of how, you know, private citizens can go out and do this sort of thing. You can't arrest everything. All right, so that's my presentation. I don't know if we have any time for questions, but I'd be happy to.
Let's thank Dr. Goodin, and then maybe if there's a little bit of time afterwards for questions.
Yeah.